I'd like to introduce our speaker. Dr. Florent Trouillas is an associate professor of cooperative extension at the Kearney Agricultural Research and Extension Center, located in Parlier, California. He is also part of the UC Davis Department of Plant Pathology, and he specializes in fruit and nut crop pathology. Today, he's going to be speaking on diagnosing and managing almond canker disease. So now I'm going to go ahead and pass this over to Florent. You can go ahead and share your slides.
Okay, well, good afternoon, everybody. For the introductions, I'll also precise my locations. I am also based in the heart of the Central Valley. I am located at the Kearney Agricultural Center, just south of Fresno in Parlier, but I'm also affiliated with UC Davis. And so I am a professor of cooperative extensions, and today I will go over field diagnosis and management of canker disease, almond canker disease. It's a major issue. And so my talk will be sort of sharing a lot of field illustrations, a lot of photographs, pictures from field cold farm visits, as well as research information from various research projects. So when we talk about almond canker disease, we want to distinguish what are fungal canker disease, just as illustrated in these pictures. So almond fungal canker disease are all these various diseases that affect the wood and the bark of trees. You can see here wedge-shaped discoloration, irregular discoloration. But in general, when canker disease start to affect an almond trees, this will result in a lot of gumming. You will see gumble developing on scaffold branches on the main trunks. And eventually, canker disease can lead to complete dieback of tree, death of trees. And eventually, if it happens or if it affects young orchards, this may lead to the death of tree and removal of young blocks and young trees. So I try to, again, orient my talk with a lot of pictures that illustrate field symptoms, because one major issue out there is field diagnostic and trying to get PCAs and farmers to really understand what's going on when they see gumming on a tree. Obviously, among the various canker diseases that I will be talking about, there's a lot of overlapping symptoms and a lot of confusion, oftentimes a lot of misdiagnoses in the field that leads to taking management approach that are not often appropriate if the diagnostic at first was missed. So again, field diagnostic is very critical in order to adopt best management practices for managing canker disease. So over the last probably 6, 7, 8 years, my lab has visited many almond orchards throughout the states. And we also conducted surveys in California to understand what are these main fungal pathogens or related pathogens associated with canker disease. We, by now, at least visited 150 orchards throughout the states. And so what appeared to be the most prevalent canker disease is ceratocystis canker. And I will provide more detail about each of the specific diseases. Following ceratocystis canker, one of the main issue also is band canker, or this disease called by the Botrytis fungi, band canker or Botrytis canker. We have seen also quite a bit of Phytophthora canker, aerial Phytophthora, which seems to be on the rise, especially with wet conditions like this year. So Phytophthora canker also is an issue. And then finally, more sporadically, eventually more in the northern part of the states, in the Sacramento Valley, we are finding things like Cytospora canker and also another pathogens also important, stone fruit in general, cherries and plums. But we also see things like Eutypa canker from Merced and north. We may find things that are related to Eutypa. So when we work to understand these few diseases that I've mentioned, it's very important also for us to understand the causal agents that are associated with each specific disease, because this will be very important when we develop strategies for management, where we test fungicide in the lab or in the field to really know what specific pathogens are involved. And so the complexity of this disease is illustrated here. Although, ceratocystis canker is caused by one main fungal pathogen, things like band canker, Botrytis canker, is the result of a diversity of fungal pathogen within this Botrytis family, right? So for U-type I canker, one pathogens, again, Botrytis at least up to 10 different fungal species involved in this disease complex. And then when it comes to Phytophthora canker, same thing. We have up to at least 4 Phytophthora species that were recognized to cause aerial canker. And same thing for Cytospora canker, at least 4 species identified within almond. So let me now go into more detail about all these specific disease. And the first one here is ceratocystis canker. Ceratocystis canker is called by, again, a fungal pathogens known as Ceratocystis destructans, formerly known as Ceratocystis fimbriata. And that's a disease that is typically associated with shaker injury. At the time of harvest, shaker eventually may damage the bark on the trunk, leading to split bark or broken bark. And these are sites of infection for Ceratocystis pathogens. Interestingly also, and that's oftentimes where confusion comes in the field, is that Ceratocystis canker can also infect pruning wounds. So these are illustrations of different cuts made on almonds or near the trunk, and this sort of specific gumming pattern that's associated with Ceratocystis. So, again, a disease that can infect at shaker injury on the trunk or at pruning wounds near the trunk or main scaffold branches. But you will notice here again, one tip here for field diagnostic is when you have infection with Ceratocystis on a tree, you'll see that gumming usually occur at the margin of the infection. So you don't have gumming throughout the infection, but always at the margin. Same thing here, look at the pruning wounds and you can see the gum balls occurring at the margin of the infection, quite typical of this disease. So this disease actually can be quite severe. I just mentioned infection at pruning wounds. This is the illustration of one specific block near Merced County, where in reds, every cells here are trees, and in reds are all the trees infected with Ceratocystis canker. So this main canker disease of almond can be quite severe in some almond orchards. So differently from many other fungal canker disease, Ceratocystis is quite unique because it moves around with insects. Okay, when this the bark on the trunk is broken and the fungal infection occur, the fungus will produce these white mycelial mats behind the broken bark with fungal fruiting bodies that will form, and these fungal fruiting bodies bear a little droplet in the tip that's ingested or picked up by a sap-feeding beetle or drosophile fly and moved to fresh fruiting cuts. Here is an illustration of fresh cuts that we have made for trials at Kearney, and within just a few hours or day, you could see how here in that case, hands are just so well attracted to pruning cuts. So again, remember that Ceratocystis is a very unique fungal disease as it moves around to infect new sites, new trees by insect transportation. I mentioned 2 important canker diseases, Ceratocystis first, then band canker, also quite prevalent in orchards. These are symptoms on young trees, on trunks. Generally, the disease will come near cracks that occur from like this fast growth on trees, excessive growth. And why we call it band canker is generally you will see this sort of a band of gum balls forming around the circumference of the trees. In very severe cases, you can see 1, 2, 3 bands forming on the trunk. And in that case, eventually tree may die. So band canker, again, is a disease of young tree, 2 to 5 years old trees, vigorous cultivars, and eventually orchards that are receiving excessive amount of water and nitrogen. And so they're growing so fast that cracks are occurring and infections is occurring at these sites. So that's the disease, band canker, also caused by Botrytis feriaceae, and so also known as Botrytis feriae canker. Band canker is usually favored by trunks that are getting wet through the use of sprinkler irrigation. So whenever a sprinkler wet the trunk, is never too good of a thing for disease. It can lead to more severe case of band canker. Final words about symptoms. Again, they often occur at the base of the trunk. They could be confused with other things, but they are generally forming in a band. And although these disease symptoms may be alarming, a lot of trees tend to recover from band canker after fifth leaf. So, after 5 years or so, a lot of trees tend to recover. And if you see at the injury or the discoloration inside, they're usually, contained with new tissue, new xylem tissue, and new bark tissue growing out from old infection. So again, the disease may be quite alarming at the early stage of the orchard development, but trees usually are sort of safe from band canker past fifth leaf. Similarly, as I mentioned, there are quite a lot of overlapping symptoms from this canker. What we see with band canker is just not only gumming on trunks at cracks, but it is also infection at pruning wounds that leads to these bright orange gumballs, and you can see here the canker developing in the bark. If you make a cut section through this canker, you will see now the typical V-shaped, weight-shaped discoloration associated with this Botrytis feriaceae fungi. So that's for band canker, again, a disease of trunks and scaffold branches and main branches. Eutypa, it's a rare disease eventually of almonds, but there have been several cases where we have diagnosed the problem. And again, the problem will often be diagnosed from Merced to the heart of Sacramento valleys and Central Coast. The Eutypa canker is caused by Eutypa alata. It's a well-known pathogens of stone fruits, apricots, and grapes. And again, it can develop at pruning wounds on trees made from scaffold selection. But typical cases where I've encountered Eutypa alata were these cracks at the junction of the main scaffold branches and the trunks. Oftentimes, these scenarios where you see a little gummings occurring at these cracks, oftentimes these scenarios were linked to Eutypa alata and the Eutypa canker. Just a few words on Cytospora. It's probably the most sporadic disease here for California, but it may occur. In that case, you will see again infection at a pruning wound and this sort of elongated canker going up the scaffold branches. So not so much of a big problem in almond. It may be present, but definitely a huge issue for plum and cherries. So if you have an almond orchard eventually next to a prune orchard, you could be dealing with Cytospora canker. So now let's get back to field diagnostic when possible and try to understand how these diseases look different and what are some of the key characteristics to diagnose these issues in the field. We will go over Phytophthora canker later in more details, but when you have an infection with Phytophthora canker, which I placed here because a lot of time there is confusion when gumming occurs on the tree. A lot of PCA have tendency to blame it on Phytophthora before thinking of these other diseases. But so with Phytophthora, you will notice gumming occurring throughout the infected area and generally near the tree crouch and making the way up into the scaffold branches. But again, gumballs throughout the infected area. With Ceratocystis canker, when you have this issue on the trunk, you will notice that the gumball only occur at the margin of the infection, okay? With Eutypa canker, I just mentioned, you will see usually young trees, a crack at the tree crotch, and a little bit of gumming. And again, with band canker, remember the name band canker will be a band of gumball going around the circumference of the trunk. So these are, good tips for recognizing this disease in the field so far. Again, here in other illustration, classic case of Phytophthora cankers where you see gumball throughout the infected area at the tree crotch, and then Ceratocystis canker on the main scaffold branches where you can see some mechanical injury here, and again the gumballs occurring at the margin only. For those who may be interested, or for diagnostic lab who may be interested with molecular tools, we had a CDFA funded project where we developed specific molecular primers for each of these pathogens responsible for canker. And so this has been a sort of a great turnout that now you can basically diagnose this disease from just sampling bark or damaged wood tissue, putting that into for DNA extraction into a PCR, and basically having a turnaround for diagnostic that's reduced to 24 hours compared to the classic 3 weeks that it will take for a lab having a morphological or by a conventional isolation approach. So just some information here to say that some molecular techniques and specific primers are now available for faster diagnostic of these diseases. So, so where do these diseases come from in almond trees, right? What are the infection codes for this canker disease to infect the trees? Obviously, all canker disease require a wound, and probably the most important infection site for a lot of this disease will be pruning wounds right near the trunk that are made for either primary or secondary scaffold selection. So really important site of infections are pruning wound made for primary and scaffold selection, primary and secondary scaffold selection. Finally, I just mentioned harvest shaker injury, but that will be a site of infection only for Ceratocystis canker and not any other disease. So if you have a shaker damage and you see gumballs, you are dealing with Ceratocystis canker. And finally, maintenance pruning. When trees are getting older, when lower limbs start to die, growers have tended to do a cleanup pruning or maintenance pruning, removing large branches. And these also can be a site for infection. Really never we see the issue from topping or edging almonds. So these young shoots that are top or edge are not any major issue for canker pathogens. These are illustrations of several of these diseases: Botrytis, Ceratocystis, Eutypa, and Cytospora. And in every cases, you will see that disease has initiated at a pruning cut. So that tells us, in terms of disease management, pruning cut eventually, when pruning is done at high risk periods for infection, pruning cuts may be protected with fungicide or biocontrol agents, and I'm going to get into more details. So again, some general biological characteristics for this canker disease. Again, except for Ceratocystis canker that is moving around with insects, most fungal pathogens move around during rain events. They're either rain splashed or dispersed by wind, but typically they will need water from the rain to hit this dead wood on trees or dead wood stores outside of orchards. And the inoculum will be dispersed following the rain, and the occurrence of wind will be dispersed to fresh pruning cuts. And this is sort of a classic fungal classic fungal biology. Fungal pathogens need rain and wind to be dispersed. So what all this information is telling us is we're gonna want to prevent infections at any of these large cut make on trunks, because once you have an infection on the trunks, now you have your tree, you have the body of your tree being damaged, which can lead to tree death. So the idea here is to, as much as possible, maximize our efforts in the early stage of the orchard development, where primary and secondary scaffold selection pruning is done. You may consider protecting this pruning wound to prevent infection that will be very detrimental if they occur at the early stage, again, of the orchard development. Remember that all management efforts will be preventive. Once you have a canker problem on your tree, it's generally too late. There's very few options when a tree is already a disease. So again, all your efforts when it comes down to the management of fungal canker disease will have to be preventive. So again, management of fungal disease will be done by the protection of pruning wound following primary and secondary scaffold selection. This is what trees look like after they've been pruned to select 3 to 4 scaffold. And again, your efforts have to be done here so you can really prevent infection at this early stage of the orchard formation, orchard development, because once you have a canker in the trunk, tree may die rapidly. So over the years, my lab, with funding and support from the Almond Board of California, engaged into field trials to determine what product may be best to protect a pruning wound. So we work with pretty much a lot of fungicide, conventional chemical fungicide that are registered in almonds. We looked at biocontrol agents. We look at paste and sealant and run, I don't know, to this point, 6-8 field trials to determine what product worked best for protection of pruning wounds. So this is quite of a just one slide summary of many years data, looking at all this diversity of fungal pathogens and looking to find best product to protect pruning wounds. And so the result from this work is thiophanate methyl, or Topsin M, in terms of conventional chemical fungicide, was one of our best products. We have product like Quilt or Rhymes that has sort of an intermediate effect when applied into pruning wounds. You will notice that things like acrylic paint did not work at protecting pruning wound. That's something to know, because a lot of time grower will be tempted just to paint wounds. And then, but finally, more even excitingly here, we found biocontrol agents and the product Vintec, which is a trichoderma-based product, which allow great protection of this pruning wound. And so that's very interesting to have biocontrol agents. Biocontrol agents, when they're applied to a pruning wounds, they may colonize the pruning wound and have a long-lasting activity when compared to conventional chemicals. So here are illustration of I've mentioned Topsin also as a great chemical protectant for pruning wounds. Here are illustrations of the amount of disease that we find here in orange in non-treated pruning wounds, and the much lower amount of disease infection. These, again, remember, these are fungicide trials where we inoculate the pruning wounds following a treatment. So we deal with high dose of the pathogens. But basically, Topsin work great for things like, again, the Botryopsferia pathogens, the Ceratocystis. And here with this picture is a good illustration of what an unprotected pruning wounds will look like. You see the discoloration and the canker below the pruning wounds when it's not protected with the gumballs, and that's again infection with Ceratocystis canker. And you see a treated pruning wounds where you have absolutely no gumming, no disease, and just a clean wound below the pruning wounds when again treated with thiophanate methyl. I mentioned biocontrol, very exciting products to us. We've done a lot of work with funding from CDFA, funding from CDPR and the Almond boards to look at biocontrol agents and their capacity to protect pruning wounds. The idea is when you treat with a chemical product will basically remain right there at the surface of your pruning cuts and not penetrate down the wood. With a trichoderma or biocontrol agent, specifically trichoderma, trichoderma will colonize the wood and allow this sort of competition, eventually mycoparasitism, against the putative pathogens. Here are further illustration of some of these trials, looking particularly at the product Ventec, a trichoderma product. When we added a spreader sticker, an adjuvant, in that case, new film P with the biocontrol agents, we have control that was comparable to the efficacy of Topsin. So again, very exciting results here to find safe, environmentally safe product, worker safe products such as the trichoderma product being able to work as good as a conventional chemical. Here are just additional illustration where in that case we tested an application methods with electric brush spot sprayer, and again the product trichoderma biological control product work as good as Topsin. With CDFA, again, we took the opportunity to look at more trichoderma products. And the general outcome is there, quite a few products within this group of trichoderma that are providing interesting control. Again, so a good biocontrol category or trichoderma product for the management of this canker disease. Similarly, here, DPF support, we looked at a different group. There's another interesting product that came here to be very interesting for the protection of pruning wound outside of this trichoderma is something called Clonostakis. So with these efforts, we are working with these various distributors and companies to have more biological control product being registered for almond. So these are coming in, some are already registered. And again, the good news is some of these products are working as good as conventional chemical. So now that we detected some of these biocontrol agents, some of the idea for us was to how to facilitate application of this product in the orchard, right? When we typically run trials, we would work with water spray bottle, which is not anything a grower could do in terms of time consuming. But so we also, question the idea of using a backpack sprayer where we would only target the pruning cuts made for scaffold selection, and then look at electric brush spot sprayers also to have larger volumes and to target these pruning cuts. And again, remember, when you're going to use the biocontrol agents, you want to have dedicated material. You don't want to eventually use the same sprayer or that you just use a conventional fungicide. These biocontrol agents are living organisms, so you want to have material that's dedicated for their use. So lastly, we looked at the application of this material using a conventional air blast sprayers, and that's where we are now in the stage of understanding the best application strategies for this product. This is just very preliminary data, again, where we use our trichoderma and conventional air blast sprayers at 100 gallon. Again, very interestingly, we had significant disease reduction compared to a non-treated control, and we had better result than the conventional Topsin. So very promising data that this product also could be used with conventional air blast sprayer system. Few words now where we at with this. We looked at 100 gallon volume with the air blast. We're looking at 200 gallons. These are just ways to have sort of better coverage when we're going to spray these young trees that were just pruned for primary and secondary scaffold selection. The idea is to get this pruning cut wet. So any product, either a conventional product or biological control product, will receive good coverage and will have the best efficacy. So right now we're looking at this different volume to again make sure any of these compounds could be efficient, again using conventional air blast system, which is the easiest approach for grower these days. For the management of bent canker, this is a slide I borrowed from my colleague Themis Michelides. The idea with bent canker is to also have preventive sprays of trees before the disease occur. And so basically the idea where second leaf orchards are in the ground, the idea will be to come with one protective spray, preventive cleanup spray in the spring. And the work from Themis have shown that just a single preventive cleanup spray of the trunks lead to a good reduction of bent canker problems. So lately, we looked at the product rhyme of flutriafol. The company has claimed that the product eventually could help reducing this canker disease, as it is known to be systemic. Flutriafol is known as a systemic fungicide. So what we've done here is to take this claim and test it ourselves. So the way we do here, we just chemigate the Rime product into the orchards, coming through the drip system, the micro sprinklers. And we looked, and then following the spray, we had inoculated branches with canker pathogen, and then we looked at lesion size and the efficacy or the impact a chemigation of Flutria could have on disease occurrence and disease evolution. So disease growth, for pathogen growth. And so again, this is 1 year of preliminary data. We had actually quite exciting result, particularly for ceratocystis, meaning that if the Rime didn't kill the pathogen, but he allowed to have sort of a slow down of the disease development, slow down of ongoing infection. So these are things that we're looking at. What if I chemigate twice or 4 times Rimes during the growing season, which is sort of a cheaper approach to chemigate compared to use your spray rig. But we're looking now to confirm this data, or these preliminary results are quite exciting. The next thing that we looked at for the management of canker disease is, what is the susceptibility of these pruning wounds if I do a pruning early in the season, just right after harvest in September? What about if I prune in October, November, December, in January? Is there a difference in susceptibility of pruning wound depending on the time of the year that I prune the almonds? And also the other question was, for how long are pruning wounds susceptible to infection? So we ran these trials in different orchards, looking at this, using this diversity of pathogens that can cause canker. And the bottom line here, the main take-home message is, in general, pruning wounds in almond are susceptible for about 2 weeks. So that's very important in our management strategy. If we're going to use a chemical protection or a trichoderma protection, that within 2 weeks, this is the window of susceptibility, and after 2 weeks, almond pruning wounds are generally not susceptible anymore to fungal pathogens. We looked at the various months of pruning, and we noticed that January pruning, meaning so a late pruning in the season, also this period of susceptibility of this pruning wound was now reduced down to 1 week. So late pruning is also probably a best approach to limit infections or the risk of infection by this canker pathogen. But basically, late pruning combined with one-time application of a pruning wound protectant immediately after pruning should guarantee great protection and great preventive approach against these canker pathogens. Scaffold selection, again, I mentioned, is very important to select branches that are vertically spaced in the orchard, so you avoid this cracking. So strong attachment with 45-degree angles, scaffold branches, is also critical to avoid any cracks formation, because any cracks formation will lead to a further disease occurrence. Just now, I'm done with the main topic, fungal canker disease, but I like to share things that we're seeing also these days in orchards. One is this Neoscytalidium canker, and it's sort of a whole rot too, because infection initiates at whole split. And these pathogens, from initiating infection at whole split, Neoscytalidium in the Botryosferia family, also cause this large dark streaking or cankers inside the wood. Very different from what whole rot with rhizopus will do, but we had quite a bit of cold these winters where growers will notice this sort of gumming and darking with black, really dark nuts remaining attached like mummies. And so if you notify this, you may be dealing with this Neoscytalidium whole rot and canker issue. Silverleaf is something that we have encountered, especially in the Central Valley, Gustin area. Once in a while, we see this really broadly widespread discoloration from basically the rootstock all the way up into the scaffold. This is caused by a basidiomycete Condrostrium purpureum. And main symptoms in orchard are illustrated by this leaf that have a lighter color. That's why the name silver leaf compared to darker healthy trees. Okay, so you may be dealing with a Condrostrium or a silver leaf problem. And again, good control has been shown before by Dr. Jim Adoskevich about using Trichoderma products or a rally as wound protectant. Wood decay, a few words. When we have windy events, windy times like these days, you may suddenly find a lot of trees falling in the orchards. A lot of time, this problem may be due to secondary infection following crown gall by things like Ganoderma. You may have heard the name. These are wood decay or hot rot. Trees suddenly are falling, and when you look inside the wood, you will find this typical sort of soft tissue, white mycelium, typical of wood decay. And wood decay again are these fungi that will form this sort of bracket fungi, bracket mushrooms. So these are typical again of things like Ganoderma, Tramilis, or Schizophyllum, and they are quite common in tree species or fruit trees anywhere. Okay, now a few words on Phytophthora. I mentioned Phytophthora is within this sort of complex of aerial canker disease. Phytophthora are not true fungi. That's why we keep them a little bit here separately from the first part of my talk. They are oomycetes. They are water molds. They are fungi that require generally water to germinate. And so typically Phytophthora pathogens will affect tree at these pockets at the junction between the main scaffolds, branches, and the trunks. So if all these branches come here together, this little water pocket may receive water, and these spores of Phytophthora that are blown up into the air following harvest and all this wind movement land on the trees and eventually are washed down this tree crotch, which leads to, again, a severe infection, severe gumming that can be diagnosed in the field by the presence of this gumming at the tree crotch, but really profuse and diffuse gumming in the almond trees. A classic Phytophthora also will be cases where you have infection making its way up the trunk in young trees. This is often the case where the bud union is buried. So supposedly your bud union, your bud line should be above the soil line. And when mistakes are done at planting, when the bud union is too low and touching the ground, touching the soil, this is where you may be dealing with severe cases of Phytophthora. Few words here for Phytophthora. We had many atmospheric river this year, and so these are rare events where we can see infection in almond trees of Phytophthora syringae. So if you prune in January and then you had this 1 or 2 waves of atmospheric river, orchard may have been exposed to Phytophthora pruning wound canker. Rarely also we see this Phytophthora occurring at just twigs without any specific wounds or obvious wounds. So an aggressive pathogen, Phytophthora syringae, quite rare that will occur every El Nino or every wet year in California, but we had a few cases this year. So for Phytophthora management, and I think I will stop my talk here unless Cheryl allow me for a few more minutes, but I think this may be good time to stop. But management of Phytophthora is basically done by water management, especially when we're talking about the root rot. But for this canker that occur above ground, it's really about keeping that bud union above the soil line. Too many times we see mistakes where bud union is buried. Proper scaffold selection to avoid the formation of this pocket is also critical. We hear good words from phosphate products also for the protections of almond trees against Phytophthora. And then you have other chemical product registers such as mefenoxam and more recently oxathiapiprolene from around this product, which help reduce the inoculum that's present in the soil, but again, some of these products also may have some systemic activity. So I may stop here, Cheryl, or do you think we have more time?
We have I think about 12 questions that have come in. So if you want to stop and answer some of these questions, we can go ahead and do that.
Surely. Okay. So you want to read the questions, or will I can read them right
Yeah.
now? Okay.
Yeah, I can read them to you. Okay, so one question that came in, are smaller trees more susceptible to the ceratocystis infection at pruning cuts? Because relatively speaking, the ratio of damaged area to undamaged area is large. Or is there something about young trees where they cannot mount an effective response?
You know, you may see ceratocystis canker in young trees because they have received more of a drastic pruning, right? So some young trees have received this primary round for scaffold selection, second round for scaffold selection. So it's not uncommon at all to see ceratocystis canker where trees have received pruning wounds. Older trees generally don't receive as much pruning, so that's less probably chances for the disease to occur. However, any trees, any age, if they suffer either shaker injury or a pruning wound, will be susceptible to ceratocystis canker.
Okay, the next question that came in, were the trials using trichoderma natural or artificial infestation experiments?
So in all these trials, we make the cut, we spray the products, and then we inoculate. So remember that we work with high spore inoculums, and so we're sort of way above the normal disease pressure. So yeah, even though we're finding good result for this product, we are working with sort of high disease pressure, high inoculum levels.
Okay, thank you. So on the Vintech, let's see, some of them are bouncing around, actually.
Yeah,
Will Vintech work on grapevines?
that's where this product was first developed in Europe, and he has been testing here also in California, and he also on grape as well has shown a really promising results.
Okay, and then the next one is, how about dipping the pruning lopper or shear into the solution?
No, this is not anything practical anyway. I mean, so a lot of time when we have this question is, are pruning shear responsible for a transmission of canker? But in general, if you cut through a canker and then back into clean wood, you don't get disease transmission. So really your best way to apply this product is with any form of spray, either manual or air blast, but you will get a much better coverage if you spray rather than try to apply it through the loppers.
Okay, and there was a little confusion. I think it might have gotten answered, but I'll go back to this one. So somebody was asking for clarification on the hand application of 0.5. I think that was the Vintech. Is that as effective as the fan at high rates?
So for the fan, when it's manual, we have great result. When you have either a spray bottle, a manual backpack sprayer, we got good result at 0.5. When it comes to using a hair blast, we have better result at higher rate. So that's 1 gram at least per liter. And using also larger volume of water for having a better coverage of the tree.
Okay, the next question, did you apply Dormex prior to the study, or did you do the trial without applying it?
There's no Dormex in our trial, and I think I don't see the relevance of this question, but Dormex is just something related to dormancy. So no, we didn't use it.
Okay. Okay, often almond orchards are next to pistachio or walnut. Are they also prone to be infected?
So when your orchard is next to almond and pistachio, one disease, especially I would say walnut, one disease to be on the lookout for is Botrytis. So Botrytis pathogen have a broad host range, and they are very common in walnut, sometimes in pistachio. And the difference is with walnut, for example, they will colonize succulent tissue, green shoots, fruits. And so there's a lot of inoculum generally in these orchards. So when you have a walnut orchard next to your almond, pruning wound protection is even more critical because inoculum is in the area or in the vicinity of orchard.
Okay, and there's another question. If an almond orchard is known to have Phytophthora, how long should one replant back into almonds? Should you choose a different rootstock from prior almonds?
Usually Phytophthora comes with a water saturation event. So like right now we have orchards that are flooded for weeks. So these are the event that really trigger Phytophthora infection. I would not, worry too much about this sort of pre-plant efforts. There are rootstock, and I don't have all in mind about which one are best for Phytophthora, but we just had a publication from UCNR about almond rootstock and their tolerance to Phytophthora. But in general, Phytophthora come from sporadic events of either having over-irrigated the orchard or having the orchard flooded due to some weather issues.
Okay, there is a question asking, what about Arondis gold?
There's maybe Arondis gold and Arondis maybe just a slight variation in the product formulation, but they are based on the same active ingredients. So if one is effective, the other one will be effective as well. I'm not just exactly sure of the subtle difference between the two, if there are any.
Okay, this next one is just a comment. I noticed you mentioned Spur Shield. I read a paper that Spur Shield tank mixed with fungicide helped reduce canker diseases.
I'm not sure if I mentioned Spur Shield in my talk. No, I mentioned the tank mix was with New Film P. Yeah, so it's from Miller Company, I believe, but same one that do Spur Shield. But the New Film P was the spreader sticker adjuvant that helped for better efficacy of this trichoderma product. New Film, the Spur Shield alone didn't show great efficacy when we tested it on almond pruning cuts.
Okay, and then there is a question came in, which lab could do molecular diagnosis, and then what would the cost be?
So, I mean, in general labs, private lab can charge anywhere from $75 to $120 per samples. You have free service from the university. My lab offer, support to farm advisors and PCA, so you can reach out to your farm advisor or PCA for getting support from our labs. I know ALNL near Davis, I think in the Dixon area is a very good lab that does a lot of molecular testing. It's called ALNL, but there are quite a few labs around the valleys that do testing for these issues.
Okay, do the same concepts apply to pruning wounds in walnuts?
We've shown that some of these products also help to protect pruning wounds with walnut. They help at preventive infection. The problem with walnut is they have such of a large pith where it's so much easier for fungi to colonize the wood of walnut. Nevertheless, this product, Topsin, Rime, also have help protecting pruning wounds when applied to walnut.
Okay, there's another one. Someone said with small trees pruned heavily in January this year, and they did not apply any protectant. Is there anything they can do now to help the trees from this wet year?
Yeah, so the next option, let's say if you're going to fear Phytophthora infections at pruning wounds, there are preventive options that would help will be to have a phosphites application when the trees will be fully leafed out during the middle of April. You can, as a preventive approach, give it a shot of phosphites. If you were to suspect any gummings, eventually then in that case, Rime, which is fertigated, may be also an alternative to slow down any potential infection.
All right. And then another question was, can you speak a little on what causes foamy canker and the effects? It is a growing problem.
Yeah. Are my slides moving here?
Yes.
I have just a couple pictures here of foamy canker. It was in the talk, okay. Foamy canker, what we came to a conclusion that foamy canker is actually sort of a secondary physiological response of almond trees that have suffered either an important Phytophthora infection. So this tree here with the foamy cankers, they were previously affected by aerial Phytophthora. We've seen trees developing or evolving into foamy cankers if they have received herbicide injury. I think my slide before is an illustration of herbicide injury. This is glufosinate injury on trunks. Eventually, these older sort of abiotic issues on almond may turn into foamy canker. So from my personal experience, I don't see foamy canker as an independent disease with a specific causal agents. I believe it's more a secondary physiological response to the tree to other issues that occurred before.
Okay, and then one more here. Are copper sprays in the dormant time still effective?
No, coppers have not shown any good result for canker in general. So they've sort of been dropped out from these experiments. Copper has never really helped for fungal canker disease. Now, copper may help for like the bacterial issues, but not against fungal canker disease.
All right. And then there's just one final comment. I'm not exactly sure if I'm going to say this right. So it says also the neostaldium. I'm not exactly sure what that's.
Yeah, neostaldium, maybe the comment came from, I've talked a little bit in my talks. Neostaldium may be, again, more of an issue in almond orchards if you are located next to walnuts. Neostaldium also causes the branch wilt disease in walnut. And when it occurs in walnut, that fungus will produce tons and tons of spores inoculum. So if you have your young almond orchards planted next to walnut, you could be dealing with neostaldiums also.
All right, thank you. Okay, so I think that is it. So thank you so much for your presentation. Thank you for answering all the questions that came in.
Thank you, everyone.
All right. Thank you, everybody, for participating today. We're going to go ahead and close out. We appreciate your attendance today, and we hope you'll be back at our next one. Have a great afternoon.